So hello, everyone. It's my pleasure to introduce today's speaker, Dr. Natividad Gomez-Roman. She is a senior lecturer at the Strathclyde Institute of Pharmacy and Biomedical Sciences at the University of Strathclyde, Glasgow. And her research spans a broad spectrum from basic science to translational biology. During her doctoral studies at the University of Glasgow, she made significant contributions to the understanding of how CMYC regulates RNA polymerase 1 and 3 transcription. And her research focuses mainly in glioblastoma, and which is an aggressive tumor of the brain, as you may know now. And she's aiming to identify certain vulnerabilities in this type of cancer to improve the effectiveness of radiation therapy and reduce the patient's toxicity. And she has developed the 3D culture models that replicate the glioblastoma clinical behavior, which are now used to screen different drugs in vitro, so we can identify some candidates for potential clinical use. So welcome, and very happy to have you here. And the floor is yours.
Perfect. Thank you, Norma. Thank you for inviting me to give this talk today, and thank you for sharing as well for the introduction with you. And thank you for listening today. I'm really honored to give this talk about the work that I have developed over the last 15 years. So, as you heard from Norma, the main focus of my lab is to study glioblastoma, which is a primary brain tumor that exhibits, as we know, a very resistant phenotype, and these tumors resist treatment and inevitably recur. And because of this, we are looking avidly to find novel treatments because these are urgently required to improve patient outcomes. And as you may know, one of the defining hallmarks of glioblastoma is their inherent chemo and radio resistance. And this treatment resistance has been attributed to a subpopulation of cells within glioblastomas that exhibit a stem-like phenotype, referred to as glioblastoma stem-like cells, or GSCs. And this is relevant for my talk because these are the cells that we're interested in targeting and we're interested in maintaining in culture, so we can actually identify those drugs that will kill these cells and maybe induce to better outcomes for patients. So here in this slide, I'm showing you the treatment evolution for glioblastoma. So as we know, glioblastoma is currently treated with radiation, temozolomide, and surgery. And this treatment followed a progression over from the 1980s to 2005s. And since 2005, the STUP protocol has been adopted by worldwide. And we know that it does improve patient outcome, but the patients inevitably will recur to their disease because they become resistant. We also know that there have been many clinical trials that have been testing receptor tyrosine kinase inhibitors such as EGFR and other kinase inhibitors using, for example, regorafenib, as well as anti-VEGF therapies, which have already received approval by the FDA. However, these treatments haven't really improved the overall survival for patients. And despite promising results in the laboratory using preclinical models for glioblastoma, we really haven't been able to achieve any improvement in the clinic, suggesting that the molecular targeted agents that we have tested so far in clinical trials have failed to improve glioblastoma patient outcomes. In particular, for example, bevacizumab, we know it's been adopted by some countries, for example, in Japan, but here in the UK, bevacizumab is not indicated for the treatment of glioblastoma because of that lack of overall survival. So that prompted us, when I was a postdoc in Professor Chalmers' lab, which a lot of this work stemmed from, to try to understand why were we failing at identifying targets that would then translate into effective treatments in a clinical setup. And for that, what we decided to investigate is what about our preclinical models is lacking, that we are not able to recapitulate the responses that you might generate in the clinic. So I decided when I joined Professor Chalmers' lab to investigate what were the current models at that time. This is taking me 15 years back. So most of the preclinical studies were being carried out using cells that indeed they had been derived from patients, but they had been grown in culture for many, many years. The cells were grown in flat surfaces, in plastic plates, and this actually changes the morphology of cells, as you can see in this image. You can see that the cells actually become much more flattened. The cytoskeleton is completely different. Here I'm showing you F-actin in red and the nucleus in blue. And you can see it's almost like that of a fried egg. So this is not how the cells would be, their morphology in the tumor in the patient. The other thing that we know is that cells were grown with media formulations that would not represent the conditions that are the tumor cells exposed to in the brain. And these cells that have been growing for several years in culture that would trigger genetic drifting of the cells. So were these the reasons why these conditions were basically not allowing us to identify those targets that will result in clinical effectiveness? Are these the reasons why we are failing at the clinic? And based on that, we decided to generate new models based on the dimensionality of the cells, and also considering conditions that have already been proven by other investigators to improve the phenotype of the cells, to maintain that stem cell compartment, so population within the cultures. So we decided to compare our 3D conditions between our 2D models, and we chose two different three-dimensional models. The first one was using scaffolds. This is an image here, which let me put my laser marker. Yeah, my laser pointer. You can see here, this is the scaffold. It's a commercially available scaffold that was developed by the University of Durham, and they are called Alvitex. These scaffolds are 200 microns in depth, and they have pores within them where the cells grow and invade. And you can see the morphology here is much more different to the morphology in the 2D conditions. You see there are spherical cells. We see these extensions that you would actually see within a tumor. We also compared that against neurospheres because several groups were working with these systems, and we wanted to also change the conditions in which we were growing our cells, and we followed that of the neural stem cell field. We started growing them in advanced MEM. We started growing the cells in growth with growth factors and devoid of serum as well. And these growth factors, I should say, they're highly at high concentrations within the tumor. So we were trying to recapitulate the conditions identified within glioblastoma patients. And once we had this model, we need to characterize it and to identify whether the responses, the molecular targeted responses to different agents was different between the 2D and the 3D models. For that, we used patient-derived cell lines by collaborating with neurosurgeons that developed multiple cell lines from patients, and we keep these cell lines at a very low passage to avoid that genetic drifting. We grow them in the conditions that I already explained, but we also add two supplements that we know for neuronal cells, the requirements for these supplements is important for their growth. And you can see here an image of how the scaffolds look like. There are several presentations of the scaffolds. You can see at the bottom of the well, that's the scaffold. And this is made of polystyrene. The cells can embed into that. And we can also have a 96 well plate format for these scaffolds. So we decided to then characterize how the cells looked like, and we compared them against cells that were grown as tumors inside the brains of mice, on tumor-bearing mice. You can see in the upper panel, these are orthotopic models. And we could observe that there was still expression of the stem cell markers, such as SOX2, in both the mouse models as well as in the scaffold system. We could also see that heterogeneity because we can still see GFAP staining in both the mouse model as well as in the scaffold. We also identified that the way that the cells were growing was very similar from in the scaffolds as that on the orthotopic models. You can see here they form in this particular cell line, the G7 cell line. We see them forming clumps, whereas in the E2 cell line, we see them dispersed throughout the brain. And it's exactly how they grow as well in the scaffolds. They don't form clumps. So they are recapitulating the growth and morphology and phenotype observed in mice. We could also see and demonstrate that these tumors have these extensions called microtubes, and these microtubes were present as well as that observed in normal tumors. These extensions, we also know that there are several genes and receptors that are overexpressed in them, and we have already characterized that, and we can also see an increase in the expression of these proteins in the 3D scaffolds compared to the 2D conditions. The cells do not lose their tumorigenicity, so they retain their tumorigenic activity, and they have 100% penetrance in the mice. So we now knew that this was, at least the model was recapitulating some of the characteristics, the features of tumors in the scaffolds. We then wanted to investigate what about the molecular targeted therapies response. And why do we do that? Because one of the goals in the lab are to improve treatment to the treatment response to standard of care that will lead to better patient outcomes. That's one of the main goals in the lab. And how do we do this? We try to do this by combining drugs with radiation therapy to achieve tumor radiosensitization. And to do that, the reason for doing that is because in that case, we're going to be able to, the radiation therapy, the same dose of radiation will be more effective when we combine it with that particular drug treatment and using that radiosensitization. And we're hoping not to increase the normal toxicity either with our treatments. So based on this, we decided to compare the radiation sensitivity of cells either alone or in the presence of different drugs. For that, because we are interested in the radiation response of cells and their sensitization, we used in the lab routinely the clonogenic survival assay. This assay is the standard, because radiation is standard of care, the assay is the gold standard assay to assess cellular radiosensitization. And as you can see here, the clonogenic survival assay is the readout for cell reproductive death. You can see here in the video that the scaffolds are amenable for clonogenic assays. And the other thing we could observe was that these colonies, their morphology actually improved when we grew them in 3D scaffolds compared to the counterparts, the 2D plates. In the 2D plates, the colonies were not as clear. They were much more, they were not as punctate. And in this case, it's a lot easier to quantify and identify what is a proper colony. So using that system, we compared then the intrinsic radiosensitivity of our cells grown either in 2D, in the plastic form, as routinely done in many labs, against the scaffold system. Here I'm showing you data from 3 different cell lines, patient-derived cell lines, and as you can see, the intrinsic radiosensitivity doesn't change. So the surviving fraction is the same at the different doses of radiation, and the curves basically follow the same line. So what about response to treatment when we combined drugs? So either drugs alone or in combination with radiation. And we focus our studies using erlotinib. Why did we choose erlotinib? Because erlotinib, we have seen in preclinical models, people have published that erlotinib has an effect. It actually has an anti-proliferative activity on glioblastoma cells in the 2D conditions. And however, erlotinib failed in every clinical trial that it has ever been tested, either alone or in combination with radiation. So in this first graph here, what I'm showing you is the surviving fraction of 2 different patient-derived cell lines grown in either 2D conditions in the scaffold system or as neurospheres. I should say in this case, this neurosphere assay, it's the neurosphere formation assay in which we only seed very few cells. We usually seed 10 cells per well, so it's a neurosphere limiting assay in which we then allow the cells to form a spheroid from one cell. So it's not an aggregation of cells, it's just one cell or, well, 10 cells that generally generate around 3 spheroids per well. And you can see in the case of the G7 cell line, we don't see any effects, but in different cell lines, we could see that erlotinib will inhibit the formation of neurospheres, whereas we did not see any effects in the 2D or in the scaffold system. That prompted us to then investigate the radiosensitizing properties of erlotinib, because other past papers have shown that erlotinib can radiosensitize cells in 2D conditions. And this graph is showing you that in the red color, the 3D cells, the cells grown in 3D and forming colonies in 3D, the presence of erlotinib did not radiosensitize our cells. However, in the 2D conditions, the presence of erlotinib did radiosensitize cells, suggesting that our model is more representative. It's reproducing those results observed in the clinic. We also observed then this was validated not only in one cell line, but in multiple cell lines. And sometimes in one of the cell lines, for example, in this case, the G7 cell line, you see in black, the addition of erlotinib actually improved the radiation protection or conferred radiation resistance to the cells. So these results suggest, or suggested at least to us, that the ADETEX model reproduces the results of the clinical trials observed for EGFR inhibitors and demonstrate that they have a higher power, higher translational power than 2D models or spheroids. We then investigated what happened when we depleted cells with VEGF, and the opposite was observed. So in this case, when we remove VEGF from the media, we see that the cells become radiosensitized in the 3D conditions, but do not change in the 2D. And cutting a story short, what I'm going to show you is the final piece of that puzzle, and we identified that in the 2D cells, EGF and EGFR axis was controlling AKT signaling pathway, whereas in the 3D, it was VEGF who was controlling the AKT activity. With that, we decided to block AKT either with an inhibitor, MK2226, which I'm not showing, or with siRNA, and we observed that inhibition or depletion of AKT protein leads to radiosensitization of cells grown in the scaffolds. We then moved on to animal work to test whether we could use an inhibitor against AKT. We used at that point MK2206. I know there are better inhibitors at the moment. And we saw that there was a modest increase in survival when we combined radiation with the MK226 inhibitor compared to either alone radiation or drug alone. So with that, we were actually quite excited that the model would be functional for testing and predicting the activity of novel compounds, novel drugs, and maybe allow us to identify potential novel radiosensitizers. And we decided to further develop the model, and for that, we decided to do a medium throughput system using clonoscreen, using a 96-well plate that has the scaffolds at the bottom of the wells. We call this clonoscreen 3D, and this is a platform for screening drug ionizing radiation combinations. So this is a schematic of the time frame for what we do. We seed cells one day before, the next day we treat the cells with drugs at different concentrations. We incubate for 2 hours, and then we irradiate our cells with either 0 or 3 gray. Following that, we allow the cells to form colonies. As you can see here, we stain the colonies at day 14, and we acquire images, segment, and process these images. And we obtain this kind of graph. So I'm showing you here a dose response curve in which we have different concentrations of talazoparib, a PARP inhibitor, and the cells have been treated at 0 gray or at 3 gray. And this model actually allows us to compare two different things. We can see first whether there is single agent activity of the drugs against glioblastoma, and we can also detect if those cells, that interaction between the drug and the radiation reduces further the activity or the clonogenicity of the cells, and that gives us a value that we have termed a radiation interaction ratio. So if there is a shift in the curves, we calculate the areas under the curve, and that ratio between those values gives us a radiation interaction ratio. This model provides a much greater surface area in 96-well plates than any other 2D platform could be done. And that's why there's very, very few 2D clonogenic platforms in there, because the colonies, it would not allow sufficient colonies in that format. So we performed an initial screen using different DNA damage response drugs. But before that, we validated whether the screen was replicating those colony formation like we would observe in the 12-well format, and we see that it does replicate that response. We can also we developed a way of automating the colony counting, because it's in a 96-well plate. I think the students wouldn't be too happy to be counting all these colonies. And we can see that both the plating efficiency and the drug radiation interaction is very similar in the 12-well plate against the 96-well plate. We can use different cell lines, although the G7 cell line is the one that works better with the automated counting. The others are not as efficient, the automated counting at recognizing them. We also then validated further whether the radiation interaction value that we were obtaining, how does that compare to the radiosensitization, the sensitizing enhancement ratio. So we compared 3 PARP inhibitors, talazoparib, rucaparib, and olaparib, against using either the clonoscreen 3D against a full radiation dose response with different drugs. And we generated the sensitizing enhancement ratio. And as you can see here, both the radiation interaction ratio and the sensitizing enhancement ratio, there was a very good correlation between these two values, suggesting that the radiation interaction ratio predicts true radiosensitizing activity. With that, we went on to perform a screen using DNA damage response inhibitors. And this is the first screen that you can actually compare multiple inhibitors at the same time. And you can see that our data identify the ATM inhibitor ACD1390 as one of the best radiosensitizers across a panel of DNA damage response inhibitors. We also identified single agent activity, very potent single agent activity of the PARP inhibitor talazoparib. We also saw inhibition of clonogenicity with check-1 inhibitors, and this is the graph that we can generate from that. And using this ClonoScreen 3D platform, we went on to identify potential novel radiosensitizers. And for that, what we did before for selection of the targets and the drugs, we performed an experiment in which we compared the transcriptomics, the RNA-seq data from cells grown in 3D conditions between 0 gray and 5 gray. And those gave us a few targets, and we identified drugs that would target these proteins. And we tested first as well in using the ClonoScreen platform. We identified that dinaciclib exhibited a very high single agent activity in this model, followed by talazoparib, which is again shown there, as well as WEE1 inhibitors. Cytarabine was a novel compound as well that we identified using this screen. Here is the whole data for that screen. You can see we have different drug targets comparing synthesis of DNA, cell cycle, which that's where dinaciclib works. It's a CDK inhibitor. We compare those NF-kappa B, and what we observed that these were our two best candidates from generated from that screen. We then validated against a full radiation dose to see whether they indeed exhibited radiosensitizing activity. And as you can see here, the activity is still observed with dinaciclib in 3 different cellular models at very low concentrations. And this is something that we are currently investigating further in the lab. We also observed cytarabine, which is involved in DNA synthesis, which inhibits DNA synthesis. We also saw a role for it in radiosensitizing our cells, and that's another arms of the laboratory at the moment. So in conclusions for this section, I showed you that the 3D Alvotech scaffold glioblastoma model exhibits better predictive value for clinical translation than previous models. I have also shown you that ClonoScreen 3D platform is a robust method to screen for single agent and radiation drug combination activity. That screen itself identifies novel potential compounds targeting pathways including cell cycle progression, mitosis, and DNA synthesis. And I've shown you that ClonoScreen 3D platform represents an improved experimental strategy for streamlining identification of novel radiosensitizers. And we hope that it will have potential to transform the landscape of glioblastoma therapy. With that, I would like to move on to the second part of my talk, which is talking about cholesterol role in glioblastoma. As you may know, there is a lot of work, several publications showing that there is an association between high blood cholesterol with different cancers, such as breast, prostate, and pancreatic cancer. And this dependence of cancer cells on aberrant cholesterol metabolism could point to these pathways as an attractive target to treat cancer, as well as to sensitize them to anti-cancer therapies. There's also now a bulk of evidence suggesting that in glioblastoma, cholesterol is also playing a role. And we know that a defining hallmark of glioblastoma is an altered tumor metabolism. The glioblastoma cells actually switch their metabolic program to adapt to the heterogeneity of its tumor microenvironment and in response to genotoxic stressors. And if we consider that the brain is the most cholesterol-rich environment in our body, we can identify how cholesterol may play a role as well in glioblastoma growth. As you may know, there have been multiple publications in the last, let's say, 8 years that have shown a potential for cholesterol as a therapeutic target. There was this seminal paper showing that an LXR cholesterol axis that creates a metabolic codependency for brain cancers. And we need to remember that cholesterol is an essential and integral part of all membranes and an essential molecule of all cells. And cells are constantly trying to maintain the levels of cholesterol to a level that it doesn't become toxic when it's in too high concentrations, or it's not lethal to cells because of very low concentrations. There have also been multiple publications showing multiple drugs that target cholesterol, for example, in this case, statins, cholesterol synthesis, showing that there might be a role of these drugs in combination with radiotherapy and temozolomide to protect, to kill glioblastoma cells, sorry, not protect. So I think I need to go a little bit over cholesterol homeostasis and how it is regulated, because it's actually quite a complex system. Like I said, too much cholesterol is toxic, but also too little cholesterol is toxic. So cells are constantly trying to maintain the appropriate levels for survival, for growth of cholesterol within them. There are multiple ways that the cells can obtain their cholesterol, for example, via uptake of cholesterol, via esterification, via synthesis of cholesterol or efflux. And also a lot of cholesterol may be also toxic, and that can be reduced by esterification of cholesterol. So cells can acquire the cholesterol from multiple pathways. So we decided to investigate what is the role in using our system, as well as 2D conditions, what is the role of cholesterol in cells. We use the same cancer stem cell enriched conditions and the same models. And in this case, I'm showing you data for 2D conditions. We see that addition or supplementation of cholesterol does not have an effect on cell growth or proliferation. However, I have to reiterate here that we are still growing these cells in the presence of B27, which is the supplement that we add to the cells for their growth. And this supplement contains cholesterol in itself. So we are adding a little bit more, but this experiments may be, there might be a bias there because there's still cholesterol within the media. We also use the Alvetech system. And in this case, what we decided to do was to see whether addition of cholesterol, further supplementation of cholesterol could improve the radiation, would improve or worsen the radiation sensitivity. And what we see here is that in the presence of cholesterol, the surviving fraction increases for glioblastoma cells, suggesting that cholesterol is protecting cells from the radiation effect. With that, we decided to run the same screen, but now in this case, targeting pathways that control the cholesterol homeostasis within cells. And we selected several inhibitors that would target different pathways, such as efflux, synthesis, transcription, esterification, uptake, and transport within the cell, so trafficking between organelles. And we could observe that there were several drugs that indeed exhibited activity as single agents. So we see a reduction in clonogenicity when we treat them, such as ivodiamine here. And we also see effects with drugs that target the synthesis of cholesterol, these two, A1994 and U18666A. And we also saw a potential radiation interaction with the statin, lovastatin. So we also tested some additional drugs that have been shown in the past that have an effect in glioblastoma, against glioblastoma, like LXR623. So now I'm going to focus on what the effects of these drugs on our cells. And because of the uptake and that paper by Villa et al., in which he showed that there was an effect, maybe a potential effect of the low density lipid receptors, LDLR, we decided to still investigate further and validate our results further. We saw first that their expression levels of the genes is very similar in brain and in glioblastoma using the TCGA dataset, and that there is no prognostic value in the expression levels of these two particular genes in glioblastoma. So overall, the survival of patients is the same, whether the tumors express high or low levels of this gene. We also observed that, as our platform had indicated, there was no single agent activity, and there is using a different system, in this case, the InQSite, and this validated further our results. So therefore, we decided that uptake was not the appropriate way of targeting cholesterol in the cells. We then decided to investigate what about APCA1. In that paper, what they showed was that if they use this LXR623 agonist, that will induce then the activity, the expression levels of APCA1, and then that would then result in a cell death of glioblastoma cells. With that, we identified a drug that activates the transporter, the APCA1 transporter that's involved in that cholesterol efflux. And we saw that the cells do express a good amount of APCA1, both at the messenger RNA and at the protein level. Ivodiamine is actually quite toxic to the cells. You can see at nanomolar concentrations, we already see activity, and it may potentially also radiosensitize the cells in combination with ivodiamine, suggesting that this would be a good pathway to target when we're treating glioblastomas. With this data, we decided to move on to animal studies, and for that, we used the S24GFP mouse glioblastoma model, which is also a patient-derived cell line, and we treated mice with ivodiamine alone first. And to our surprise, and well, not a very good surprise, I should say, we saw that ivodiamine reduced the survival, the overall survival of mice compared to our control, suggesting that ivodiamine actually was inducing a faster growth of glioblastoma cells in the brain of mice, and that's why the mice succumb faster. We then decided to compare this against data from using the TCGA dataset, and we do see that glioblastoma tumors express much higher levels of messenger RNA for APCA1, and that it's actually high levels of APCA1 are a prognostic factor here. We see patients, there's worse survival of patients with high expression of APCA1 than patients with low APCA1. And what we think it's happening as well, it's not only that the cells, and somehow by releasing their cholesterol from and activating APCA1, we think that we're also activating APCA1 present in astrocytes. However, we're still trying to identify if this is the case. So APCA1 is involved, and it's also present in astrocytes, and astrocytes are the main cells that synthesize cholesterol within the brain and use APCA1 to transport for the efflux of cholesterol to feed either neurons, or in this case, it could be even glioblastoma cells. So we're actually investigating this further. However, this was a bit disappointing because we are seeing the opposite effect that what we wanted to do. But on the other hand, we can also see that the cells do respond to targets that are involved in cholesterol pathway. So based on our previous clonoscreen platform and our screen using the cholesterol inhibitors, We then decided to investigate cholesterol synthetic pathway. Why? Because of that lovastatin treatment, which we did see that radiosensitization. I'm showing you here the 3 drugs that we selected. So we selected statins. We also selected U1866A, which also showed potent activity in clonoscreen 3D, and AY994, which was also another of our hits from the screen. I know statins have a bad reputation, but let's still go through and see what we obtained. So the first thing we did, we tested whether the cells were expressing these genes, and they do. In fact, they are the dihydrochol. So these drugs target these particular enzymes: 24-dehydrocholesterol reductase, 7-dehydrocholesterol reductase, and the beta-hydroxy beta-methyl glutaryl coenzyme A reductase. And with that, we decided to investigate their activity both as single agents from multiple statins. In here, I'm showing you 4 different statins, and they were all exhibiting really low activity at low concentration, so below 1 micromolar. We also observed that statin and radiation treatment radiosensitizes cells, in particular simvastatin and pitavastatin were the best radiosensitizers in our studies. We then went on to assess this using a different 3D model. In this case, I'm not only seeing one cell or very few cells, we are actually allowing the cells to aggregate and form a spheroid, and we measure the growth of the spheroids. You can see that at different concentrations, so there is a dose-dependent inhibition of growth using simvastatin in 2 different cell lines. This is actually protected and rescued by addition of cholesterol, as you would expect if it was regulated by cholesterol at the lower concentrations. However, at the higher concentrations, we see that there is further cell kill. There is a reduction of the growth of the spheroids, suggesting that the effects observed here are not the same and they are not regulated by cholesterol. We recapitulate these effects using a different assay, an MTT assay. We do see that further decline in survival of cells, in cell viability at the higher concentrations of cholesterol and simvastatin. And we have recapitulated again this, not using only an MTT assay, but also using an InQSight in which we can measure the proliferation of cells in real time. And we see that they do radiosensitize cells, pitavastatin. In this case, we see an increase in the gamma H2AX foci, which is a marker for double-strand breaks. And we see an increase in the presence of 2-gray, as you would expect. The same at pitavastatin. It's a bit higher than with 2-gray alone. We also see an increase in the cells the percentage of cells showing mitotic catastrophe in the 2-gray plus pitavastatin conditions. And we also see a reduction of the percentage of mitotic cells in the combination treatment. We are currently investigating what are the mechanisms involved in this statin radiosensitization in glioblastoma cells. But we also wanted to investigate the other 2 drugs just because of the really bad reputation as well of statins, but we're still interested in statins. We can see here, this is the second drug. This drug targets the biosynthetic pathway at a much later stage of the biosynthetic pathway, so there might not be any effects like the statins, which target at a very early stage. And we see that there is increase in, potent activity of the compound, not as potent as the statins, but the statins might be due through their other pleiotropic effects at high concentrations. We also saw very interesting results that in this case, This drug is specific for glioblastoma cells. So what I'm showing you here in red is glioblastoma treated with pitavastatin. In blue are astrocytes treated with pitavastatin. We see a very strong response also from astrocytes. In green are glioblastoma cells treated with the AY drug, and in purple it's astrocytes treated with the same concentrations as we treated the glioblastoma cells with the AY compound. And you can see that the astrocytes do not respond, suggesting that this might be a very good strategy to inhibit cholesterol within the glioblastoma cells, and it may result in no toxicity in normal cells. We continue to validate this drug using the spheroid model. You can see that the spheroids do decrease their growth in the presence of AY, but we do need a bit higher concentrations to see these effects in one of the cell lines. In the second cell line, we actually saw disaggregation of our spheroids, suggesting that the cells are actually dying. This is completely rescued at any concentration with cholesterol, suggesting that cholesterol is the main molecule that it's actually having the effect where the AY9944 is having its effects, inhibition of cholesterol. And we went to validate this further using clonoscreen again, but in this case we used cells that had a CRISPR knockdown of the dehydrocholesterol reductase 7, which is the target for the AY99 drug. And we see that there is a very strong, robust reduction of colonies following the depletion of this protein. And also there is radiosensitization almost to comparable levels as those observed when we deplete DNA PK, which is a protein in DNA damage repair. We have also seen very strong effects with U18666A, and this is a molecule that targets the 24-dehydrocholesterol reductase, but it also has other targets as well. With this one, we see a very, very potent activity of the drug to cells, very, very low concentrations. We see a decrease in the EC50s, and it also exhibits potential radiosensitization in our 3D scaffolds. So to conclude, I've shown you for this part that we have identified 3 potential targets exhibiting activity on GBM cells. These are 7-dehydrocholesterol reductase, and we can inhibit that using AY9944. This is specific for glioblastomas and has no inhibitory effects on astrocytes. And I have also shown you activity for 2 other drugs, U18 and statins that target DHCR24 and HMGCR. I have also shown you that there is radiosensitizing activity with statins, U18, and with the knockdown of DCHR7. And our findings also demonstrate that for future therapeutic strategies, we must not only consider the effects on glioblastoma cells, but we always need to consider as well the cellular responses of brain cells to those treatments to find an effective treatment for glioblastoma tumors. And with that, I thank you for listening. I apologize if I went a bit over 45 minutes. And these are a list of our collaborators and the members of my team here. Thank you, and I'll stop sharing.
Thank you very much for the very interesting presentation. It's really, really interesting, all the work you have been doing. And well, now I open the floor for questions. Please go ahead if you have any questions. I don't see here. Maybe in online, no questions? So I have one question.
Okay.
It's a little bit, well, it's regarding the statins, we know they are very commonly used, but I don't know if these drugs pass the blood-brain barrier. I'm familiar with the side effects of these drugs in, for example, skeletal muscle, but I don't know if these are able to pass the brain-blood barrier, and if the concentrations that at which you are seeing, observing some radiosensitization are similar to the concentrations that we can find in a patient that is taking these drugs.
Yes. Yeah, so we know that simvastatin does cross the blood-brain barrier, and pitavastatin actually, although there's data in the literature that it doesn't. I was last weekend at the European Association for Neuro-Oncology meeting here that happened here in Glasgow, and there was a presentation there showing that pitavastatin does cross the blood-brain barrier in patients and in tumors. Most of the data that I think the main issue with the blood-brain barrier is there's a lot of information there based on mouse models. So PK studies using mouse models. The blood-brain barrier in mice is different to the blood-brain barrier in humans. In humans, it's a lot more leaky than we think compared to mice. The other thing to consider is the brain tumors might be also more leaky, even more leaky than the normal brain. There's a paper from Anthony Chalmers showing that olaparib did not cross the blood-brain barrier in tumor-bearing mice. They did not detect olaparib or detect at very, very low levels, whereas they actually taken samples from patients and that have been treated with olaparib, and they can actually detect much higher levels, much higher concentrations of olaparib in the brain of patients, suggesting that indeed tumors are more leaky than we actually considered before. So in the case of the statins, I do think at least the simvastatin, pitavastatin will cross the blood-brain barrier. And in fact, the group that presented the work on pitavastatin, they are currently running a phase 0 clinical trial in Belgium.
Okay, that's very, very interesting. Yeah, so that's important to highlight that sometimes the animal models, the mice used as animal models are not the best animal models, because some of the that we are trying on them don't actually work or work better in the human. So, and it's also very interesting, your model about the 3D and the comparison of the results that you got with the 2D cultures and the 3D cultures, because many, many people, including myself, we are conducting our experiments in 2D models. And as you've presented, it's not, but sometimes it's not representative. And also, I think it's important for reducing the PDX models as well. Like if we screen in 3D models as you're doing, and then we find good options to treat patients, we could then try in PDX models. So we will reduce the animals that we are using, like unneeded.
Yeah, absolutely. That's actually how we started the whole project for the platform. We were funded by the NC3Rs here in the UK, the National Centre for Reduction, Replacement and Refinement. And because, yes, indeed, that's what we were aiming for, to reduce the number of drugs that reach that stage of preclinical testing in animals. If we can identify only those drugs that will have the most likely will be effective in patients, we are already a step ahead. So yes, reduction of animals is also a main focus in our lab, and that's why we're developing better models in vitro.
That's great. It's very good. So do you have any questions? No? No questions? I think because most of the students that are here in the first year and they have not followed radiobiology yet.
Oh, I see. Okay.
Yeah, so they will have radiobiology in January, but I probably they will have more questions in the future.
Okay.
Yeah, thank you very much. I will contact you aside because I'm very interested in your work and I would like to collaborate further with you.
Of course, yeah. I'm always open for collaborations. Really,
Yes,
really
I'm
excited.
more focusing in metabolism, but in the glycolytic pathway, and as I mentioned to you, in lung cancer. But there's a lot also in glioblastoma about the glycolytic pathway, and I think combination maybe of those pathways, cholesterol and that could be very interesting also as for radiosensitization of glioblastoma. So I'm very
Absolutely, yeah.
happy to have this opportunity to meet you and to learn more about your research.
Thank you very much, Norma. Thanks for the invitation. It's
Thank
been
you,
an honor.
everyone, and have a nice weekend.
Thank you.
Thank you very much. Bye.
Bye. Bye.
Sorry, guys, it's a little bit difficult because you don't know the basics of radiobiology, but then when we will have radiobiology, then you can ask me and then we can contact her again. And so